Right, let's just dive straight into this explainer. Today, we're taking a strict analytical scalpel to a very specific, highly deceptive medical marketing pitch. It's a pitch you very likely encountered in your own practice or studies, beautifully wrapped up in the shiny veneer of cutting-edge research. But, spoiler alert, once we actually dig into the numbers, the data tells a completely different story.
Picture this for a second. A pharmaceutical rep walks into a packed, chaotic clinic and drops this exact quote right on your desk. This brilliant-sounding pitch is quite literally the perfect trap set by medical marketing to bypass your critical thinking. They hand you this perfectly manicured PDF, heavily highlighting just the abstract and the conclusion. I mean, it sounds like absolute magic, right? An acid suppressant that doesn't just treat the stomach, but actively protects the kidneys from severe chemotherapy toxicity, and maybe even boosts cancer-fighting effects on the side? When you're swamped with patients, it is incredibly easy to just nod, accept that beautifully formatted conclusion, and adopt the drug right into your clinical toolkit.
So let's actually look at the bait here. This is from a 2026 paper by Alanisi and colleagues. The authors present what look like miraculous night and day drops in kidney injury markers in a rat model. Basically, the rats given vonoprazan alongside the heavy-hitting chemo drug cisplatin showed these beautiful, clean reductions in key kidney injury markers. We're talking serum creatinine, BUN, and KIM1. And here is the real hook. The authors boldly attribute these incredible results to cellular-level protection. They specifically cite complex mechanisms like autophagy regulation, antioxidant defense, and anti-inflammatory actions. On the surface, it looks like completely airtight, groundbreaking science.
But here's the thing. It is remarkably easy to shatter this whole elaborate illusion of cellular protection. We don't even need a PhD in complex molecular biology to do it. We literally just need to look at the body's basic plumbing. Because when we peel back all that high-level jargon about autophagy and cellular regulation, we uncover a much simpler, far less glamorous reality hiding just beneath the surface.
Let's walk through the actual biological sequence of events here. Any first-year student knows that severe vomiting and fluid loss lead to dehydration, which absolutely tanks kidney function. While when you inject a massive dose of cisplatin into a rat, it aggressively stimulates the chemoreceptor trigger zone in the brain. This triggers intense, immediate nausea. Now, rats actually lack the physical anatomical ability to vomit, so this manifests as pica behavior, where they frantically eat non-food items like their bedding. But the physiological end result is exactly the same: massive, rapid fluid loss. The circulating blood volume drastically drops, and the glomerular filtration rate physically tanks. Long before we even start talking about complex cellular toxicity, we are looking at a purely mechanical, pre-renal acute kidney injury. So of course, markers like BUN and creatinine are going to naturally skyrocket.
And this is where the massive dehydration confounding comes into play. If we compare the paper's incredibly bold claims on one side with the simple, grounded biological reality on the other, the truth becomes painfully obvious. Vonoprazan is an incredibly potent acid suppressant. It didn't magically wrap those kidney cells in a molecular shield of protection. It simply suppressed the stomach acid, stopped the vicious nausea loop, and prevented the severe dehydration that spikes the kidney injury numbers in the first place. It is a profoundly mundane, symptom-masking effect, all dressed up in a lab coat as a cellular miracle.
You know what this really mirrors? A classic special fraud scam. In a typical fraud scam, the victim is presented with an utterly absurd contradiction, like the police supposedly mailing you an arrest warrant to help you avoid being arrested. But the perceived authority of that official-looking document just forces compliance. The medical marketing trap works the exact same way. The absurd contradiction of claiming molecular cellular protection when you merely prevented a rat from getting dehydrated is completely masked by the sheer, overwhelming authority of an international, peer-reviewed journal publication.
It's actually kind of fascinating, isn't it? The intense, daily pressure of a busy clinic, combined with the shiny label of a 2026 Frontiers publication, can so easily override our basic physiological logic. Smart, incredibly well-intentioned doctors are tricked simply because they literally do not have the time to look past the impressive formatting and that gold star peer-reviewed stamp. But escaping this authority trap is absolutely essential if we want to truly protect our patients' long-term health.
To do that, let's pivot and look at the timeline trap. The study we've been dissecting is an ultra-acute model. We're talking just 10 days of monitoring. The researchers basically looked at dying cells, desperately spiking their survival proteins over a week and a half, and they slapped the label autophagy improvement on it. But out in the real world, these drugs aren't prescribed for just 10 days. They're prescribed to human beings for months or even years at a time.
Trying to use a 10-day acute rat fire drill to justify years of continuous, daily human administration is frankly scientifically reckless.
If we want to understand the real chronic danger here, we have to look at basic thermodynamics. Vonoprazan is a highly lipophilic weak base with a notoriously high pKa of 9.3. Because of these very specific chemical traits, when it is taken chronically, it irreversibly accumulates inside the lysosomes of cells all over the body. It completely neutralizes the V-ATPase enzyme. Think of it as essentially paralyzing the cell's basic ability to take out its own trash. So, instead of regulating autophagy like that 10-day paper confidently claimed, chronic use actually induces chronic autophagy failure.
And look, this isn't just some abstract theory. When we pull actual real-world pharmacovigilance data, like the massive JADER database over in Japan, we see the devastating tangible consequences of this cellular paralysis. We definitely aren't seeing widespread kidney protection. We are seeing severe, undeniable real-world signals for acute interstitial nephritis and full-blown chronic kidney disease. That 10-day rat data basically smokescreened the actual long-term kidney damage that occurs when you chronically paralyze the body's cellular cleanup mechanisms. So, how do we actually protect our patients from this? To permanently immunize yourself against this specific brand of medical marketing, just adopt this simple two-axis framework whenever you review a new paper. Axis 1 is the data representing a short, acute scenario when the actual clinical application is going to be chronic. And Axis 2 is the supposed protection merely masking pre-renal dehydration rather than preventing true parenchyma cellular damage. Run new claims through those two simple filters and watch the illusions just fall apart.
I'm going to leave you with this final thought to really chew on for your own clinical practice. Every single time we introduce an effective drug into the human body, we demand a biological toll. It's an absolutely unavoidable trade-off. Knowing what we now know about the deceptive illusions of 10-day dehydration models versus the harsh reality of long-term cellular paralysis, you really have to ask yourself, is the true chronic cost of this marketed illusion a price you are genuinely willing to let your patients pay? Thank you so much for joining me on this explainer today. Keep asking those hard questions, stay critical, and I'll see you next time.